This is a webinar presented jointly by a number of NCRIS projects entitled Accelerate Your Translational Research, and marks the opening of the Pipeline Accelerator voucher systems. I'll just say at the outset that I see some familiar names in the attendees list, but also quite a few unfamiliar names. I think that's terrific. It's great that hopefully you're hearing about this scheme for maybe the first time. So I will start by acknowledging country. I'd like to note that TIA acknowledges and pays respect to the traditional custodians of the land on which many of us are gathered today, and I pay my respects to elders past, present, and emerging on the lands of the Yagara Turrbal people in Brisbane on the south side and north side. I just want to very quickly go through the program of the webinar today. And it's very simple. It's now a bit more simple than previous webinars because we have a lot more facilities and available capabilities. So we're going to start with a little introduction from myself on the scheme itself and a little bit about its history and its intentions. Then John Paraso, who is associated with quite a few of the projects, NCRIS projects, and we'll give a bit of an overview of the capabilities of each of the NCRIS organisations at a very high level, because as I noted, there's a lot of technical capabilities. It will take far too long to go into even some of them in detail. So John will give a bit of a helicopter view, a bit of a greatest hits summary of the kind of capabilities available within the network, and we invite you to follow up with us directly if you have any further questions or want to express your interest in applying. And finally, MJ Schwab of TIA will give a little bit of a talk about how to apply for the scheme, some things to note, some things to remember about maximising your potential success of the scheme. And just before I kick off anything else, we do have a question and answer box open. If you have any questions, please put them in the question and answer session. We'll either answer them along the way or we'll address them after the end of the seminar as well. So where does all this come from? Well, supporting all the NCRIS organisations is NCRIS itself. This is a National Collaborative Research Infrastructure Strategy. It's a long-term federally funded scheme to support the development, maintenance and access to national research infrastructure. That is the facilities and expertise that researchers need day to day that they may not want to or wish to or be able to establish in their own labs. So it's intended to be an open access network of technologies and the expertise to go with it, for the benefit of all Australians, researchers and SMEs particularly. It was established quite a few years ago now, and with some considerable public investment, and employs nearly 2,000 technical experts and researchers, facility managers, and they're all there to help researchers get their research done. Now, this is the NCRIS network in a whole bunch of logos. NCRIS serves all the sectors of Australian science, or areas in which Australia is aspirational or world leading. Everything from urban research to digital research to marine research to microscopy to imaging to public health and at the top, because it's my diagram, so I put TIA at the top, is Therapeutic Innovation Australia. But of this network, here we go, the projects that are associated with the Pipeline Accelerator scheme are these for Therapeutic Innovation Australia, Bioplatforms Australia, Phenomics Australia, and ANSTO's National Deuteration Facility. And as I noted, John Paraso will give a bit of a helicopter view of each of these capabilities and what they offer to researchers. So why vouchers? What's the idea of this? And where do we operate? Well, We are looking to help projects go from post-discovery into the first couple of steps of the translation journey, very broadly from TRLs 3 to 6. This is the so-called valley of death, where it is difficult to sometimes get the support, the funding, and the expertise needed to go from a promising discovery to something that an SME or a company or even a technology transfer office would be interested in investing in and putting a lot of time and effort in. So the intention of this scheme and the mission of quite a few of the NCRIS projects here in general is to help projects bridge this valley of death and start on the translational journey. So I'm just going to give a very high-level view of what the voucher scheme is. It is a competitive scheme which provides subsidies of up to $50,000 to offset the cost of accessing a range of NCRIS facilities for the purpose of fee-for-service work. Excuse me. So it's a subsidy. We acquire we award up to $50,000. That money has to be matched. We don't mind where the money comes from, as long as it's real. It could be from a grant, it could be from strategic funding from a university, it could be from investment, it could be from charity or foundations. What matters is that there's sufficient resource to make sure that the project happens. We say up to 50,000. If, for example, you get a quote from one of our facilities and the quoted cost is, say, 70,000, then we would support $35,000 of that, and you would need to find the other 35,000. That's what we mean by the 50% matched funding. Now, this scheme is open to Australian SMEs and academics. If there's an international aspect to your project, please come and talk to us, because there are some circumstances where those are eligible, but it is mostly for Australian-based SMEs and academic researchers. We run the scheme every 6 months, and we'll do so for the next couple of years under our current conditions. The opening date was actually last Monday morning. The closing date, and this will be reiterated a couple of times, including at the end of this seminar, is April the 17th at 5 p.m. Brisbane time. One of the key facets of this scheme is that you can get a decision very quickly. In 6 to 8 weeks after the closing date, you will have a letter with either a letter of award or a letter saying that you have not been successful, but it will be quick. And because of this, you'll see the application form is similarly fairly straightforward and simple without too much ancillary information required. Very important point is that we take zero IP interest. This is an accelerator. We are just trying to help research projects happen. We don't take any IP interest or beneficial interest of any kind in the projects. We do take a bragging rights interest, if you like. We do like to come back to our awardees every couple of years and see what the funding has led to. And in that way, we collect the impact stories and the good news stories that are very important to TIA and to the federal government to justify their investment in this scheme. And our colleague MJ will go through how to apply more of the specifics on how to apply and eligibility a little bit later on in this talk. But I just want to present some of the metrics thus far. We've been operating since 2018, and since then, TIA, as well as our 3 partners, have collectively made 332 awards, including some supplementary vouchers, which are vouchers to access Bioplatforms Australia and National Deuteration facility, in addition to accessing either Phenomics Australia or TIA's facilities. This has totaled nearly $11 million, but has enabled 10 times that much in downstream investment and successful later stage development grants. Grants for development, not development grants, unfortunately. We have a collectively 57% success rate, so it's very much worth your while to explore this possibility. And as you'll see from John's presentation, which is very soon, we have now about 75 research infrastructure facilities across our group of 4 NCRIS projects. So there's a lot there that can be accessed. One other scheme, which we also run at the same time as this, which is specific to TIA, is something called a technical feasibility assessment. And this is something that's kind of specific to cell and gene therapy facilities. It's an advisory service if you wish to go to one of our facilities and get their advice on the developability or manufacturability of a cell or gene therapy. That is also available. It's a consultancy valued at $10,000. How that works is we give the money to the facility and they give the applicant $10,000 worth of a consultancy on manufacturing methods. So that is something that's also available. Happy to discuss that individually if you wish. Just very briefly before I pass on to John, it's always nice to reflect back on where the schemes come from. And we started in 2018 with a very small number of projects, 8 awards across a couple of 6 facilities. Over the years, we've started to dedicate specific resource to the scheme. In 2020, we ended up totaling about $1.5 million worth of awards, including a special COVID round. And then later on, we reached a century of 100 awards in 2021. And we're very excited to start to include Phenomics Australia and more latterly, the Deuteration facility and Bioplatforms Australia, which is not quite on this version of this diagram, because nonetheless they have joined and it's going from strength to strength. And we are starting to see quite a few outcomes and positive good news stories resulting from these vouchers, including people coming back for a second opportunity and a second award to progress their project in a different facility, which is what we love to see. So without further ado, I'm going to pass on to my colleague, John Pariseau. And John, if you could just give me a sharp next, and I will let you know, and I will advance the next slide for you.
Great. Thank you, Stuart. Hi, everyone. Thank you for joining us this afternoon for this Pipeline Accelerator voucher webinar. My name is Dr. John Pariseau. I work across 3 of the NCRIS partners today that we'll be presenting. So that's Bioplatforms, Phenomics Australia, and Therapeutic Innovation Australia as a strategic partnerships advisor. But I will also, of course, be talking about the National Deuteration facility as well. Next slide, please, Stuart. So let's start with TIA. So as Stuart has gone over briefly, TIA is a national network of 36 facilities and counting, supporting mostly the translational research across a range of modalities that I'll show you on the next slide. Now, the investments that TIA enable can be accessed by academic researchers, and more in the last 3 to 5 years since I've joined, significant uptake by industry to progress their novel therapeutic potential products. Next slide. Thank you, Stuart. So in a nutshell, the TIA is spread into 4 specific capabilities categorized. So you can see there's cell and gene therapy at the top, small molecule therapeutics, traditional small molecules for drug discovery, biologics and vaccines, and more recently, RNA products, as the boom in RNA world, as everyone knows. On the next slide, Stuart. So here you can see a bit more detail about what's involved in each. So just to give you a quick overview, this slide that demonstrates some of the therapeutic products are usually taken orally, like small molecules and antibodies in many instances, and others are delivered by injection, RNA products and vaccines, as we know, and also regenerative or cell and gene therapy is delivered by infusion. And you can see here within each section there a quick summary of what's involved. Small molecules have some compound inventory and library storage and logistics, high throughput screening, chemistry, drug metabolism. RNA has people that can help with design and production and the lipid nanoparticle formulation. Biologics and vaccines, very, very well versed in antibody drug conjugate design and recombinant protein production from non-GMP up to GMP level as well. And lastly but not least, cell and gene therapy as well, with GMP-like and GMP manufacture of cell and gene therapies for research and clinical use, viral vector production, all the quality control you need to get into the clinic. Next. So this is a comprehensive geographic representation of TIA's network. As you can see, it's we're almost running out of colours for the facilities. It's grouped in the different categories: biologics, cell and gene therapies, RNA products, small molecules, and the additional one here that wasn't on the previous slide is our high throughput screening capabilities for the screening of molecules for translation and drug discovery and development. So pretty much a great national spread. I think the only one we can add to this network is Tasmania. And you can see the colour coding here as well, all the facilities mentioned. So please reach out to us if you need to be put in touch with any of the leads in these facilities. Next. So here's exactly what I was just saying. Here's a QR code for accessing, contacting us and how to get to these TIA capabilities through us. You can either use the information there through the info therapeuticinnovation.com.au or contact any of us directly. We'll be able to help you tailor what you need for your voucher with the right expertise and the right facility to help enable you. Next. So it's almost a little bit like a translational pipeline, the role of NCRIS partners that I'm going through today. So one step back is a sweet spot for Phenomics Australia would be the functional characterisation and molecular basis of disease using models. Similar metrics there to TIA. On the next slide, I think we've got more of a breakdown of the capabilities. So this schematic here shows you that the pipeline for Phenomics Australia is mostly in that in vivo, in vitro disease modelling and biobanking through to high throughput screening using functional genomics and pathology. Next. So if we just scroll through briefly, biobanking is particularly important for our mouse and in vitro models, cryopreservation and the databases of strains and so forth. And also importantly, they can help enable your importation of specific models mouse models, example, that you might need. Next to it. This is in vivo predominantly rodent models. So where we have used gene targeting strategies, CRISPR-Cas9 technology predominantly these days. There's also transgenic Drosophila production. You could use a Drosophila model as opposed to rodents. Transgenic mice and just enabling the construct design that you need for your in vivo engineering. Next, Stuart. More and more importantly these days, we're moving towards non-animal models as decreed by the FDA, and whilst we call that non-animal technologies, the worldwide moniker now is NAMs, meaning New Assay Methodologies, and that's all about minimising the use of animals and trying to replace as many models as possible with in vitro. So here we have organoids that are developed from patient samples or from human-induced pluripotent stem cells, and we use them for high-throughput imaging and screening, high-content imaging, bioprinting, and have a registry of the stem cells underway. Next, Stuart. So leading genotyping and phenotyping we have at the moment, but it's spread across multiple facilities, so we'll skip that one. But functional genomics and high-throughput screening I just mentioned predominantly uses these in vitro models, and we can do pooled CRISPR screening, RNAi screening, compound screening, lots of different types of phenotyping in the in vitro world, and including the bioinformatics associated, like crop-seq and so forth. And last but not least, pathology. Next, Stuart. Where you have your phenotype and your functional models, you still need to see what's happening at the individual rodent level or other animal species. So we have availability here for pathology of all the tissues within an animal's body to be analysed, scored, and the data curated, and expert analysis, as well as the slides digitally scanned. Next, Stuart. So moving along now, I guess also one step earlier in the translation process is Bioplatforms Australia. So Bioplatforms Australia is predominantly set up across, next slide, Stuart, three main mission areas. So whereas Phenomics and TIA are predominantly involved in human health therapeutics, the missions for Bioplatforms is a bit broader. It also encompasses human health and well-being, but apart from those missions, it has significant infrastructure in facilities that can help primary industries and food production, and also to help enable biodiversity and environmental issues. So the way it has these impacts, on the next slide, Stuart, is predominantly using multi-omic and bioinformatics across these three examples. So I won't drill down to too much detail, but you can see in the nature environment, there are programs concerning biodiversity and fish, fungi and birds, across to primary industries and food, where many of our projects are about increasing the yield from Australian barley, reducing animal health, reducing animal disease and improving their health, and plant pathogens and wheat reference genome. And last but not least, in the human health area, where Bioplatforms are involved in cardiovascular stem cell characterization, antibiotic resistance, and melanoma, for example, melanoma genome project. And on the next slide, Stuart. So how Bioplatforms has done that is to set up pretty much, as I was mentioning, multi-omic expertise across the nation. So genomic capability, proteomic capability, metabolomic capability, and more recently adding synthetic biology as we move towards larger scale production of enzymes and different cell types that can be produced on a larger scale for uses, not just therapeutic, but other uses in animal health and also, as I mentioned, in plant. And last but not to exclude bioinformatics, which we all know is very important to be able to analyse the amount of data that everyone is probably aware coming out of all these multi-omic areas is enormous. Bioinformatics will be increasingly important with the AI and machine learning underway, as we all know. Next slide, Stuart. So this is just to follow the sequence of national footprint. You can see, once again, Bioplatforms spread across those categories of capabilities, spread across most of Australia, and it's quite a strong presence in WA, where there's quite significant genomics and metabolomics capability as well. And more recently, synthetic biology there in the red, where we have some initial nodes in plants in bio, but also Genome Foundry at Macquarie University and IDEA Bio up at UQ. Next, Stuart. I just wanted to mention here, so you can contact myself or you can contact Sarah Richmond, my colleague from Bioplatforms here. But as Stuart mentioned earlier, Phenomics Australia and TIA are the primary voucher enablers, but more recently, supplementary voucher applications could be made in conjunction with those primary vouchers for Bioplatforms and for the National Deuteration Facility, which I'm about to go next to. So, the National Deuteration Facility, I'm presenting this on behalf of my colleague Karen Weil. They have been with the Pipeline Accelerator for several rounds now, and I'll just go into what is deuteration on the next slide. Oh, sorry, before I do that, I'll go into the actual facility. It's an Australian Nuclear Science and Technology Organisation research infrastructure platform, and it's also NCRIS co-funded. There's some animation on here, I think, Stuart. So the NDF custom make deuterated and stable isotope labelled materials for your drug development programs. Excellent. And the way they do that is to provide deuterated or isotope labelled molecules to researchers. They can also chemically and biologically isotopic labelling and help investigate the molecular structure and function of the enhanced properties, the properties you're trying to enhance in your drug development program. Next slide. Yep, I think I mentioned that. We can go to the next slide on actually what is deuteration. So just to take a step back to Physics 101, many moons ago for me, the hydrogen atom typically has a proton and electron. Deuterium atom has the neutron added and making it a heavier atom. So why we use deuterium or deuteriate molecules is on the next slide, Stuart. So it's used as a characterization tool. So deuteration of organic molecules can make the drug or the in development, the candidate drug in development more stable. You can use the neutrons for analysis and neutron scattering. It also aids with nuclear magnetic resonance spectroscopy to get a better idea of structure. It can be more easily identified using mass spectrometry and infrared spectroscopy as well. Just to list a couple of obvious advantages of deuteration. Next slide, Stuart. So yes, here's a slide here demonstrating what I've just mentioned. So the neutron scattering can help you elucidate structure with under X-ray once you've added the deuteration to the molecule. And next slide, Stuart. So with NMR, you can discriminate between the deuterated and non-labelled isotopes with the labelled isotopes, so they can help you with elucidating the structure and the optimal structure for drug development in many instances. Excuse me. Next slide, Stuart. Mass spec. This is a little bit out of my wheelhouse, but it obviously does help you identify what your drug once deuterated, can get metabolized to within the body, for example. And last but not least, the deuteration and substitution also helps determining kinetic effects, measuring the kinetic effects with enzyme rate limiting effects, and also binding rate constants and so forth. So on the next slide, Stuart, just to summarise that the NDF capabilities I've just mentioned are available through the Pipeline Accelerator scheme as a supplementary voucher. That we can produce bespoke deuterated or stable isotope molecules, get multiple labelling of proteins with different combinations of isotopes, and it will help with the quantitative analysis as I just described. And I think I've got on the next slide, Stuart, who to reach out to apart from us. No? Yes, okay. Yes, so reach out to NDF inquiries, have a look at the website, but by all means reach out to one of us. We could put you in touch with Karen or any of the other colleagues we have at the NDF that we're able to discuss a potential application with you. So that's all for partners who are in the Pipeline Accelerator voucher scheme for now. It's always changing, it'll be different, I'm sure, next year. But I'll hand over now to MJ, who will go over exactly how you apply and how to increase your chances of being successful. Thanks, everyone.
Thank you, John. So I am MJ Chua, the Director of Programs at TIA, and I'm going to talk to you about how do you apply for a Pipeline Accelerator voucher. Next, please. So let me start off with minor tweaks we introduced in this round and also reminders. And the first one, so the new, what's new in this round is that we have assigned DOIs to every award for recipients to acknowledge us in publications. So starting this round, we will include a DOI in the award letter. We've also assigned a DOI to every award, every voucher awarded in the past, and they can be found on the outcomes page on our website. Next, please, Stuart. And this is a screenshot of the outcome page on our website, and you can find the DOI on the far left of the table. Next, please. So supplementary vouchers, or Stuart spoke about supplementary vouchers before. So these vouchers are intended to complement primary vouchers, and therefore supplementary vouchers cannot be the main focus of your application. So if you want to apply for a supplementary voucher, you must also apply for a primary voucher to access either a TIA or a Phenomics Australia facility. And the value of the supplementary voucher must not exceed the value of the primary voucher. Next, please. Applicants seeking access to providers outside applicants' research or business units will be prioritized. And next point. And this is a reminder for previous recipients. So you are welcome to apply for additional voucher, but completion report must be submitted. Otherwise, your application will not be considered. Next, please. Again, this is a reminder. So avoid last minute submission because it often affects the quality of your application. So our voucher round opens every 6 months. So if you can't make it to this round, you can apply in the following round, which is only 6 months away. So I would advise you to allow enough time to engage with provider of interest. And next point, please. And co-develop your application with a chosen provider. It would be great if you can share the final version with the provider before submission. And the last point is the word limit. So just remember there is a 400 words maximum. So this is not a NHMRC or ARC grant. So we're not asking for a very long application. So we really appreciate if you can keep your responses very focused and straight to the point and keep within the word limit. Next slide. So I'm going to talk about what we're looking for. The new projects to providers, the next one. Applicants seeking access to providers outside applicants' research or business unit. So the reason being, this voucher scheme is designed to encourage access to facility outside the applicant's research or business unit. So if you're going to access a provider that sits within your research or business unit, you must demonstrate that voucher funding is needed to enable access to this facility that would not have occurred otherwise. And the next one. As Stuart mentioned, we encourage applicants from previously awarded projects seeking continued support or expanded access to facilities, techniques, or services. And the next one. We also want to encourage applications seeking access to a combination of TIA, Phenomics Australia, NDF, and Bioplatforms Australia to support the same project, which means that you can apply to access multiple facilities in the same application. So for instance, you can access Compounds Australia that is supported by TIA to get compounds, and then you do your screening at Monash Functional Genomics platform, which is supported by Phenomics Australia. The next, please. So the examples of therapeutic projects supported include develop novel assays for identification of potential products. Next, generate key data to support the next development step, or strengthen the case for further grants funding. Optimize and develop potential therapeutics, including de-risking projects by solving technical problems. And manufacture high-quality materials to support preclinical and clinical trials. So this is a non-exhaustive list. So just any therapeutic projects, but we don't really have any particular preference. Next slide, please. And this is my last slide, and I'll walk you through the application process. It is very, very simple and very straightforward. And the first step is to approach provider of interest or providers of interest if you are planning to access multiple providers on the same application. So if you're not sure who these providers are, you can check out our website or scan the QR code next to it. So you can see here there's QR codes for primary, supplementary, and TFA providers. So you can also find the facility contacts on these websites. So discuss your project needs with the provider or providers so they can help you to understand what services are available, what's feasible, and what fits within the scope of the voucher. So they were able to guide you in shaping your application so that it aligns with our voucher requirements. So once you've had this discussion with the providers, you can start preparing your application using a template provided. Just a reminder that our application form is an online form, which means that it cannot be saved and returned to at a later date. So please use the template provided, which you can download from our website, and the QR code is next. Yes, there's a QR code. Again, I strongly encourage you to work very closely with the provider as you prepare your application. And the final step is submit your application. So the applications close on 17th of April to 5 p.m. Australia Eastern Standard Time. And the next, this last slide. So yeah, so that's all for the info session today, and thank you for your time. If you have any questions about the voucher scheme, please reach out to us. You can contact us at info@therapeuticinnovation.com.au. We're here to help.
Thank you very much, MJ, and thank you very much, John, for that. If any attendees like to put their hand up or answer a question live, I think there's a little button here which is allow to talk. So if anybody would like to ask something, please let me know. Otherwise, you can just contact us online and we'll do everything we can to help you identify, craft, and submit an application to this scheme. Other than that, it reminds me to say thank you for your attendance. I hope to see some new applications from some new projects. Always looking for novelty. But again, we are also, at the same time, we do welcome repeat applications for the same project as it moves down the TRL pathway. And with that, I'd like to bring this webinar to a close, and thank you very much for your attendance this Thursday afternoon, wherever you're dialing in from. Last thing, this has been recorded, and we will post this on our website as a bit of a record of this event, and to note some of the changes and the minor updates that we've presented. So thank you very much, and I'd like to wish you a good afternoon.
Thanks, everyone.
Thank you.